welcome to Tales of the Resistance, a podcast about antimicrobial resistance. On today's show, as always, we are joined by a rotating lineup of members of the I Am Responsible Project, a nationwide team of educators and researchers working on solutions to antimicrobial resistance. I'm Beth, one of your hosts today, and I'm joined by Mara.
Hey, everybody.
And Amber.
Hello.
Last episode, we hosted Professor Jean Mclain, a research scientist from the University of Arizona. I highly recommend checking out that episode, as well as our video of Professor Mclain presenting her fascinating research into reusable wastewater and antibiotic resistance. Today's episode will be something of a post-episode talk about our takeaways from our talk with Professor Mclain. Mara, do you want to start us out? Anything that stuck out to you or questions you still have?
Yeah, the one thing I meant to ask her, I saw that she was on this paper that was doing work in the Arctic for, she was talking about establishing that there are a lot of native resistance genes. And she talked a little bit about like the pristine environments that she had found AMR in. And I know she was on a paper that was working in the Arctic where they found resistance in Arctic settings. And so the results wouldn't have been that different than what we did get to talk about, but I just would have loved to hear about what it was like to do research in the Arctic.
Yeah.
All of the places that you get to find, when you read those papers, like they found it in soil and Mount Everest, or they found it
in a deep, dark cave.
Somebody had to go there and collect, we're going
who goes there
tomorrow, right? And we're going to be sampling in a cornfield in Nebraska, but
exotic.
And I'm like, gosh, I've got all this logistical stuff. I got to figure out how we're going to get our probes there and our people there and all of the sampling equipment that we need. And
that's just
we need to do some research where you're doing soil sampling like in Egypt or someplace
organize that.
Really test my organizational skills. This going 2 hours away to a field in Nebraska is not cutting it.
But
just saying the scenery is a little different.
Yeah. Well, this was a really interesting podcast, getting to hear more about the environmental side of the One Health perspective and hearing more about water, wastewater, and groundwater. And I just find that really interesting. It's one of my more interesting parts of AMR, I think. So,
have you ever talked to Dad about your recent discovery of water science?
No.
As an interest area?
That would be kind of interesting.
Our dad is a water scientist, Amber.
Oh,
and,
yeah.
That is true. I didn't even think about it as the
same. We worked on his projects and had dinner with him, every day for 20 years, and we're never interested in his science. This is exactly the sort of thing that Beth was like with my AMR talks at dinner table.
Oh my God, it's so true. I could not focus because it was I didn't have a back. Okay, I don't know if this happens to you, Amber, but the glazed eyes, the glazed mind, if you don't have enough to connect things into in your head, then it's just all like threads that are hanging. It's like, a spider web where there's like nothing to connect it to. Threads hanging in the wind, like they have nowhere to connect to.
Yeah.
I'm just like, I don't get this. But now that I have some background information, I'm able to follow things.
Yeah.
So
I still feel like that sometimes, like when she starts talking about metagenomics and DNA, like I just barely understand. And
metagenomics, I didn't know, and I was going to ask now for clarification, but I forgot.
Well, we have talked a little bit about this, but probably not with that.
Andrew did his presentation on it.
Yeah. So like in our class, we talked about how, there's the difference between the bacteria and the DNA. And a lot of times the research that has been done, because of the costs of exploring DNA in the past, they would just look at specific genes. So if you've heard of somebody talk about quantitative PCR, or they just start talking about quantities of finding genes in an environment, that's been the sort of most common way to examine DNA for a long time. And the reason is that you're looking at just a very small portion of the overall DNA in a sample, and you use methods I don't need to explain here in order to only amplify that. And that would really reduce the amount of time and money that would be required. But now with the technology, improving, and also a big part of this is data science that is behind being able to identify all of the different very small subvariants of an individual gene and still say, yes, that is that same gene, because otherwise the little ones will get just overwhelmingly confusing for manually like going through and looking through them. But now with the combination of new data and new technology that is improving the speed of DNA replication, you can take a sample and just look at all of the DNA in a sample in a full string, and that gives you the so it's called metagenomic data. So it's like genomic, so all of the genes from a species all together in the series that they were in the original cell, and then it's meta, so it's everything together.
Yeah.
And that means that we can now not just say there's this many of this particular resistance genes in environment, but we can say we have this many of this particular resistance genes that are attached to this kind of other resistance genes that are in this species of bacteria. And so you have all of these components together that can say, what are the risks? I mean, it's kind of getting towards the idea of you have a lot of resistance in non-pathogenic species. So what is the should we count that? Should we care about that? Or we have a lot of these genes that are traveling together, like a resistance that is to iron toxicity is.
So you have the whole picture.
You have a much bigger, more complex picture of how the species and the genes are interacting within an environment. We don't do metagenomics in our lab. We don't have the capacity. We don't have the machines, and it also is you get the actual scan, but then there's the whole process of like decoding what the scientific, because the data comes out as basically molecules, these molecule combinations in letters, and you have to take that and go from that to genes. Like, what does it actually mean?
How do you know this?
I mean, we did study it, and I would love, like Andrew and I have talked about it a lot, like this is where we want to go. Like if we really wanted to study microbiology, this is what you would want to know about.
Okay.
And like I looked into a program one time of doing my PhD in basically bioinformatics and metagenomic analysis. There's a lot of work that can be done in that field, and you would never be short of a career, but it is a very lab intensive. You're in a microbiology lab all the time, or on a computer working on analysis for the results, but it's a much less varied field, I feel like, than some of the things that we do. And I'd love to dabble in it, but I don't want to spend the rest of my life doing metagenomic analysis and just every day all long. But it is very interesting, and like
it sounds really interesting.
Yeah. So I'd love to do it one day. But we'll have to find a project that we can just do it for a little while, like in somebody else's lab, and they can tell us how to do it, and we can kind of get a little of the insight, but we don't have to do it all the time.
We're not married to it.
Yeah, that's my dream goal, like sort of.
We want a mistress, not a wife.
Metagenomic mistress.